So in today's lesson, we're going to look at errors in meiosis. So we've spent a few days going over the ins and outs of meiosis, that is sexual reproduction, a process that produces gametes. But we haven't yet talked about things that can go wrong. So typically when we talk about things that can go wrong in meiosis, we're talking about it happening either during crossover, where there's an exchange of DNA between homologous chromosomes, or during independent assortment. So that's when chromosomes are moving to either end of the cell. So, nature generally takes care of this because typically the gametes that results as a result from this process where there are errors, those gametes will die. And so they will never become a part of a viable zygote that will go on to become a human being, or if we're talking about other species as well. So, however, if they do survive and are fertilized, then that means that every single cell in that new organism is going to contain the chromosomal error. So let's take a look at things that can go wrong. So first off, I want to talk about, generally speaking, errors that are caused by changes in the chromosome structure. So typically that's what we're talking about when we're looking at crossover during prophase 1 of meiosis. So there are two things that typically can happen. The first of which is that the chemical bonds during crossover don't form properly. Now, we typically just sort of think about this as being, okay, well, just different colored portions changing, but you need to remember that that really we're talking about molecules of DNA, and so there might be problems with the type of bonds, be it covalent or otherwise, that are going on during that exchange. It might not properly reform during crossover. Now, another thing that can happen typically is that non-homologous chromosomes accidentally crossing over. Now, typically, crossover will happen between chromosomes that are homologous. That is, one maternal chromosome and one paternal chromosome that would be homologous, and they would be exchanging genetic information. So, for example, maybe the little, red section and blue section that have switched, maybe the genetic information in that portion relates to eye color. But the problem is that if you have non-homologous chromosomes crossing over, then you're not switching the same type of genetic information between those chromosomes, and that leads to error. So what I'm going to have you do at this point is to pause the video and look for a worksheet on Google Classroom outlining the different types of errors that we see that could occur during crossover. And what you'll learn about are some genetic disorders that are specifically associated with this type of error in meiosis. When you finish the worksheet, you should come back to the video because I'm going to talk about the errors that are occurring during independent assortment. So in the next part of the conversation around errors in meiosis, I want to talk about changes in chromosome numbers. So this typically happens during the independent assortment stage of meiosis, and it can actually happen during meiosis 1 or meiosis 2. And so I thought I'd start by talking about the diagrams first. Now, I want you to keep in mind that we're only showing one single pair of homologous chromosomes, but of course, if we're talking about humans, we would have 22 other pairs in here, but for simplicity's sake, we're only showing one pair. So this could happen with a single pair of homologous chromosomes, or it could happen with more than one pair of homologous chromosomes. Okay, so in a process that we call nondisjunction, what can happen is instead of chromosomes moving to different poles in the cell, they might move to the same pole accidentally. So in the diagram on the left, this really is the errors occurring during meiosis 1. And so what's happening in meiosis 1, normally you would see, if we're isolating one homologous pair, we would expect one of the chromosomes to go to one pole and the other chromosome to go to the other pole. But for some reason, there's that non-disjunction error where they do not separate properly. And so what you end up with is you end up with one cell with twice the number of chromosomes and one with less, half the number, right? So, let's just do a quick count here. So, I'm remembering that I'm starting out with 23 pairs here, right? And then I should end up with 23 and then 23 over here. Remembering, of course, that when I say 23 pairs, I really mean 46 chromosomes, right? But what actually happens in this type of non-disjunction is instead of getting 23, I'm getting 24 over here. And instead of getting 23 over here, I have one less. I would have 22. So you can see that, it still adds up to our 46 chromosomes. It's just that they're in different cells. And so for that one homologous pair, both of them go to one cell and neither of them go to the other, hence the difference here. And so when they split during meiosis 2, I have some gametes that have an extra chromosome, and I have some that have one fewer chromosome. Okay, so now in the diagram on the right, what I have is this is where the error happens during meiosis 2. So in other words, I have everything going perfectly fine during meiosis 1, right? I start off with 23 pairs, and 23 pairs, and then half of them are going here and half of them are going here. So I still have, let's count that, I still have 23 chromosomes in meiosis 1 after meiosis 1. Everything has happened normally there. However, in meiosis 2, where the centromere splits, for some reason, the centromere doesn't split as it normally should in one or more cells. This is another non-disjunction that occurs. So perhaps in one cell, the centromere splits properly, and I end up with a sister chromatid in each cell here. However, with the other cell, the centromere does not split, and so I end up with that chromosome moving to one gamete and none of that chromosome moving to the other. So in this case, one gamete would have one extra chromosome and the other would have one fewer. So there's no problem if this happens and the gamete that is eventually fertilized is, let's say, this one. But there is a problem if the gamete used for fertilization that produces a zygote is this one, this one, this one, this one, this one, or this one. Those are all problematic because the recipient of that gamete, when they're looking to have their full complement of 46 chromosomes, will either have extra chromosomes or less chromosomes than they are intended to have. Let's write a little bit of this down. So when we talk about sister chromatids not separating properly during meiosis, what we're talking about we can either be talking about the case where it's happening during meiosis 1 or meiosis 2. That process, however, is collectively referred to as non-disjunction, and the disorders associated with it are referred to as non-disjunction disorders. Okay, so instead, one cell will get both sister chromatids and the other will get none. So the result, as we spoke about, is that some gametes are going to have too many chromosomes, while some will have too few or too little. They'll be missing some. So now I want to introduce you to a couple of different terms that are important and are associated with non-disjunction disorders. And they are monosomy and trisomy. So let's use our background knowledge to try and figure out what this might mean. So mono we know is a prefix that means one, and tri is a prefix that means three. So let's look at this. So monosomy refers to a situation where the cell has lost a chromosome due to non-disjunction. So in other words, instead of having the appropriate number of chromosomes, they might have one or more less than they're supposed to. Okay, so if they've lost one chromosome, that's referred to as monosomy. So instead of, let's say, for instance, in their own complement of chromosomes, where, on a karyotype, you might see, pairs and then smaller pairs and so on and so forth, but you might hit a spot along the line where you see a single chromosome and then nothing where there's supposed to be a pair. So that is what, on a karyotype, monosomy would look like. Trisomy, on the other hand, as you may suspect, is a case where instead you have an extra chromosome. So, for instance, to relate this to what we just talked about, so if I was looking at a karyotype of a situation where there is trisomy, then again, on my karyotype, I might have, my pairs, my homologous pairs listed, on and on and on. And somewhere in that karyotype where I should have another pair, instead of just having two, I might see a third. So there you go. So monosomy, trisomy, both caused by nondisjunction or a lack of separation of chromosomes at some point in the process of meiosis. So let's take a look at some chromosomal abnormalities caused by nondisjunction disorders. The first you're probably familiar with. It's referred to as trisomy 21, but trisomy 21 is the name given to what is commonly referred to as Down syndrome, right? So Down syndrome is a trisomy on the 21st pair. So on the 21st pair of chromosomes, somebody who has Down syndrome would have an extra chromosome present. So instead of 2, 3. So that has a frequency of about 1 in 800 live births. And I think we're fairly familiar. Most of us have someone that we know with Down syndrome. But that typically will result in varying degrees of intellectual disabilities, some different palm creases. Their eyes are all what we call almond-shaped, flattened face, short stature, and those are just a few characteristics. So there's a perfect example of nondisjunction that we are all familiar with that you may not have thought about. There are several others in your textbook. I'm not going to go through all of them, but I am going to point out a few. I'm going to scroll down to when this happens with your sex chromosomes. So we know that the 23rd pair of chromosomes determines our sex. So trisomy 21 is an autosomal problem condition. So it doesn't affect male or female. In fact, it affects male and females equally. Okay, so now going down to sex chromosomes. So it can happen where you, instead of just having an X and an X chromosome or an X and a Y chromosome, you have a third chromosome in that set. So you would have an X and a Y. And so this happens in about 1 in 1,000 males. And you might be saying, well, why are they considered male? Well, anytime there's the presence of that Y chromosome, that particular individual would be considered male. So that's referred to as Klinefelter syndrome. And what you would see some sexual immaturity, so maybe the inability to produce sperm. You might also see some sort of breast-like structures. And that's one possibility. So XYY, back in my day, I want to say they called that the super male syndrome, but I haven't seen that lately in our resources. And again, happens in about 1 in 1,000 males, which is a fairly high frequency. But that's referred to as Jacob syndrome. So there was this misconception that there were a higher than average number of individuals in the penal system and the jail population that were super males or that had this XYY chromosome. But that myth has been since debunked. But they may be taller in age, and that's, again, in association with the extra Y chromosome that's there. I want to talk about the triple X syndrome. So again, but it's less frequently that we see this, where you would have 3 X chromosomes, as you would expect, because of a disorder relating to the sex of the individual, that you might see some conditions associated with that become irregular, like the menstrual cycle. And lastly, so all of these I should mention are examples of trisomy, right? And you have 1 extra chromosome. It just so happens that it's a sex chromosome. But the last one here, X and O, and the O just symbolizes that this is a monosomy, that they only have a single X chromosome and no second sex-determining chromosome. So this is far less frequent in occurrence, but it's referred to as Turner's syndrome. So the individuals are typically short in stature. They might have a webbed neck, and they might, again, as you might expect, be sexually underdeveloped. So if you're curious about any of these disorders, feel free to search out some reliable sources on the internet, if you're curious to see and to learn more about them. But they certainly are interesting examples of non-disjunction. So I hope that you were able to make sense of today's lesson, and that if you have any questions, that you will reach out to me. You should expect to see a gizmo shortly that will ask you to work through some a virtual lab on karyotypes involving errors during meiosis. So look for that soon.